Okay, I guess I'll start. Hello everyone, I'm Sudiksha, and today I'll be discussing about the contents of week 8, and also have some old assignment questions which I'll be discussing. Um, Let me just share my screen. Is everyone able to see the screen? I'd assume yes. Okay. Last week we had discussed about the endomembrane system, which consists. Am I audible? Can someone let me know if I'm audible or not?
Yes, ma'am.
Okay. So last week we had discussed about the endomembrane system, which includes organelles like the endoplasmic reticulum, the lysosomal compartments, the plasma membrane, the Golgi complex, etc. The nucleus is also a part of the endomembrane system. uh, so if this is basically a more detailed map of how the endomembrane system of the cell is spread. Traditional diagrams, you would see that the endoplasmic reticulum is sort of a very compact structure which is localized to the area near to the cell, near to the nucleus, uh, which is not which was the uh, which was the opinion for a long time, but recently papers have shown that the ER is not constrained to the place near the nucleus. Rather, it forms a network throughout the cell, just like the microtubules or the actin filaments form a network throughout the cell. Um, so there are areas which are like continuous sheets of ER, and there would be areas which form tubular networks of ER. Uh, this ER has associations with different uh, different motor proteins, with microtubules, other organelles, which basically helps it to regulate the traffic of proteins that it synthesizes. Uh, one so these are basically some of the proteins that form that are associated with the ER. This blue thing that you see here is the Golgi. The Golgi, however, is a very compact structure as compared to the ER. And when you perturb the cytoplasm, if cytoplasmic the cytoskeleton, if you remember last week, if you perturb the cytoskeleton, you see that the Golgi sort of dissociates. It becomes spread very much similar to how the ER is spread in a normal cell. However, the structure of the ER is like this in normal untreated cell. Um, Again, this is another beautiful image which shows how the ER is spread through the it's sort of interweaved through the microtubule network. So the green filaments that you see are the microtubules, and pink or purple ones that you see are the ER structures. Now coming to the nucleus. So nucleus is the is found in almost all of the eukaryotic cells except for red blood cells. Uh, that's another story. So basically, red blood cells in their immature state, in their erythrocytic state, have nucleus. However, they lose the nucleus for other benefits. So the red blood cells do not have a nucleus, whereas every other cell in the body has it. Uh, so if you were to see how the stiffness varies in a single cell in any part of the body, you would see that within the whole cell, the area which contains the nucleus is the stiffest. It has the you'd need to apply the most pressure there. uh, and you can easily observe the nucleus in a light microscope. You would see um see the cytoplasm and nucleus only when you see the cell through the light microscope. You can't see other organelles. For other organelles, you need to go for more advanced methods of microscopy. Uh, Again, average mammalian nucleus has a diameter of 6 micrometer. If you consider the cell having a diameter of say 12, 30 to 30 micrometer, it can be anything. Yes. Sorry, did anyone have any question? Okay. Okay. Uh, and the the nucleus is the it occupies around 10% of the total cell volume on average. It can differ. While I think there would be an image, but anyway, while so if a cell is migrating from one place to another, you'd see that the nucleus is the last part of part of the cell that would migrate. Also, if the cell has to migrate through a mesh of say ECM or extracellular matrix, uh, the maximum force that a cell would feel would be because of the nucleus, because it will basically jam the cell. So it you deforming the nucleus becomes very difficult during migration. um if you look at the structure of the nucleus, it consists this if you consider this is the nucleus. The outside of the nucleus is a double membrane uh, nuclear lamina, which is nuclear membrane. There is a double layered nuclear membrane. There is the nuclear lamina, the nuclear plasm, which contains euchromatin and heterochromatin regions, which we'll be discussing further. The nucleolus, which is basically the core of the nucleus. Uh, nu- Again, nuclear envelope or nuclear membrane. there would be ribosomes sort of scattered on the surface of the nucleus, and this sort of the outer nuclear envelope becomes continuous with the endoplasmic reticulum, the rough endoplasmic reticulum to be specific. The smooth endoplasmic reticulum would be sort of farther away from the uh, nucleus. The surface of the nuclear envelope is also littered with these holes, which are called nuclear pores. They basically help in the traffic of things in and out of the nucleus. Um, so this is how what a interface uh, nucleus looks like. It's all uh, it's all uh, condensed, and you can't basically differentiate between two different chromosomes. In However, these the DNA is always wrapped around these structures called histones, which are proteins formed up which are protein complexes formed up of 8 subunits. These The degree at which these histones are able to wrap the DNA around it, the yes. Is there a question or something? Um, I'm not sure about the exam date. Sorry. That would be something that is something that would be given to you by the professor. Okay. 29th October. You have your answer. Okay. So, okay. Uh, so depending upon how the DNA is wrapped around the histones, there will be different degrees of compaction of the DNA. There are heterochromatin regions inside the nucleus. There are also euchromatin regions. These heterochromatin regions are more densely packed. They are more compact, and so if you see the structure of the heterochromatin, they have a so these uh, this there is this model called beads on string model, wherein so basically when you visualize these histone bound DNA, you would see them as beads which are woven through a string. So that's where the beads and string model comes. So if you try to measure the dimensions of the structure through that, you would see that the heterochromatin has a repeating fiber of 30 nanometer, whereas the similar structure is reported to be 11 nanometer in case of euchromatin. The euchromatin, as compared to heterochromatin, is very loosely packed. Uh, it it basically enables the DNA to be able to be exposed to the RNA polymerases or other proteins that are floating around uh, uh, inside the nucleus. So the genes that need to be expressed very frequently, those genes would be present in the euchromatin region of the nucleus. and not in the heterochromatin region. The genes in the heterochromatin regions are usually silenced. Um, this uh, so as I said before, there is a the nuclear envelope is a double membrane layer, and there are these holes that are peppered throughout the nuclear envelope, and these holes are called nuclear pore complex. And there are many reviews which show what the components are in, and you can look at it if you want. There are other proteins that are also associated with the nuclear membrane, and I don't think you need to know all of this. The link complex, however, is something that links the cytoskeleton that is present inside the nuclear membrane to the cytoskeleton that is present outside of the nuclear membrane. So basically, the cytoskeleton present outside is the uh, actin cytoskeleton or the microtubule cytoskeleton, right? This link complex basically forms a tether between that cytoskeleton and the nuclear cytoskeleton, which is mostly composed of lamina. Uh, so there is this thick layer of lamina uh, uh, near the nuclear envelope, which sort of provides support to the nuclear envelope, for otherwise it would collapse. Uh, the lamina is also associated with the chromatin, uh, different forms of it, and uh, there have been multiple reports as to what other proteins that are present in the envelope do, but I think you won't need it. Uh, this so the This is EM that basically shows that the nuclear pore sort of the nuclear envelope sort of merges into the rough ER, and uh, ribosomes are present in both nuclear envelope and the ER. Um, yeah. So, basically, the uh, the nuclear pore complex forms a donut kind of shape at the nuclear envelope, and there is a transporter inside, which is usually used to transfer RNA from the that is synthesized inside the nucleus to the outside of the nucleus towards the cytoplasm, so that it can be received by the ribosomes, and then proteins can be synthesized from that. Uh, there are multiple proteins which make up this structure. Um the this is basically the linker between the nucleoskeleton, so to say, and the cytoskeleton, the link complex, which is composed of plectin, nestrin, sun 1 2. I don't think you need to remember these names, but still good to know them. uh, Depending upon what uh, uh, proteins are present in the link complex, it can link to the intermediate filaments present in the cell or the actin filament. uh, Usually it is linked to both of them. And the sun 1 2 complex would link this Nesprin plectin complex to the lamin, uh, the nucleoskeleton. Yes. uh, Yeah, this has a list of all the proteins that are present in the nuclear pore complex. A lot of diseases have defects in these proteins, and this basically leads to disruption of the nuclear structure and the traffic of things between the nucleus and the cytoplasm. This is a beautiful image that shows how assembly and disassembly of the nuclear pore complex. So during division, almost all of the components of the cell sort of disintegrate, they dismantle, and then after the division has occurred, they reassemble again. And this is basically a depiction of that. uh, and Yeah. There are different kinds of lamins found throughout the nucleoplasm. Humans, two major kinds of lamins: A type, which is lamin A and C, and B type, lamin B1 and B2. These A type lamins are present in terminally differentiated cells uh, such as intestinal cells, such as lung alveolar cells, skin cells, things that do not are no longer going to divide. Uh, B type lamins are however present in all type of nucleated cells. They are present in differentiated cells. They are also present in the stem cell population of the body. Um, Mutations in these lamins will lead to different kind of diseases, and one of the examples for that is the Hutchinson-Gilford progeria syndrome, or progeria, which is a defect in lamin A, if I'm not wrong, gene. So because this gene is mutated or absent in the baby, the baby has advanced aging or premature aging. uh, okay, that's about it. And so up Even inside the body, there are different stiffnesses that the cells encounter. Cells in the brain would encounter stiffnesses in the range of 100 pascals, whereas there are cells that are present inside the bone which would encounter stiffnesses of up to 1 gigapascals. And depending upon what sort of stiffness the cells are in, their behavior would be different. Their migratory abilities will be different, their shape would change. So there have been a lot of studies that show that if you put the same cells on different kind of different stiffness platforms, using different kind of methods, they would have different shapes, different morphologies, uh, their nuclear sizes might change. They might have different migratory capabilities, all of that. um, so there is a continuous crosstalk between the what the substrate is and how stiff the cell is, and this in turn leads to a different uh, regulation in the stiffness of the nucleus, because as I said, nucleus is the most stiffest part of the is it's the stiffest part of the cell, and it is sort of what blocks the cell from migrating uh, very tightly packed situations. um yeah, this basically shows that if the cell has to move, it would move in such a way that all of the organelles, all of the contents of the cytoplasm that are present uh, uh, present in the cell except for the nucleus would first move forward. The nucleus would be the last thing. So when the cell would be migrating, the nucleus would, you can see that the nucleus is at the following edge of the cell instead of the leading edge of the cell. uh, Again, the substrate stiffness would depend upon what the substrate is, what is the cross linking of the fibers that is in the substrate, which if you are talking about extracellular matrix, what is, how fast it can be degraded or modified, how is the viscoelastic response of the cell and of the substrate. And depending upon that, the cell is going to modify its contents, like it would rearrange its actin cytoskeleton, its microtubule cytoskeleton. It is going to rearrange how the even the nucleoskeleton, its the linkage between the actin cytoskeleton and the lamina, uh, will lamins would be different, all of that. So basically, it all boils down to what kind of environment the cell is in. Also, depending upon how much, uh, you perturb the presence of lamins in the cell, it would lead to different activities of the cell. If you perturb the presence of various nucleopore complex, it would affect the cells differently. So basically, in this scenario, the nuclear lamina has been depleted somehow, which basically leads to very wobbly kind of cells. These cells don't uh, are not as stiff as cells would be with a normal lamin. Yeah. So basically, there is a continuous crosstalk between how the nuclear envelope, how stiff the nuclear envelope is, how the lamins are arranged, and how they are interacting with the actin, and all of this affects how the gene expression would be, what the nuclear shape is going to be, or what the stiffness is going to be, how it's going to develop. It would also affect the division of the cell. uh, it affects how the cells are going to migrate. uh, it has been found that if the cells are left on a very stiff substrate, they are going to migrate faster, whereas if they are going to be left on a very soft substrate, they migrate slow. uh, the crosstalk is also going to affect how the cell is polarized. So in cases such as the intestinal cells, there is a apical side and the basal side. So in those cases, the cell needs to be polarized some way, and even during migration, the cell needs a certain direction to it, and all of this is a result of the crosstalk between actin and nuclear envelope. um, there are different receptors that are present on the surface of the cell. Some of the there are pathways which are mechanosensitive, and all of these so these receptors would basically sense what the substrate stiffness is, what the environment is, and they would likewise make changes inside the cell, and so that the cell can survive in that scenario. uh, one uh, one of the things that the actin cytoskeleton sort of plays role in is also the cell division. So during cell division, as I said, all the components are going to disintegrate. They will be present, but they will be present in a very disintegrated form. um, for example, the nuclear envelope is going to vanish. The thing, yeah. Anyways, the nuclear envelope is going to vanish. The ER is going to be fragmented. Golgi is going to be fragmented. All of that. So this this is basically what mitosis is, which is when you get when you divide one cell into two identical daughter cells. And this is what happens in case of say skin cells. When you need to replenish skin cells, there are certain stem cells at the base of the skin, and these would duplicate and give rise to new cells. Similar things happen in the intestine and in the lungs as well. There is another kind of cell division called meiosis, which is what is used to make the germ cells, uh, the sperm and the egg. That is something different and hasn't been discussed in this course. Um, but anyways, the the cell cycle, so to speak, has been divided into four major uh, portions. There is the mitosis or M phase, which there is the interphase, which is basically the phase where the cell grows and accumulates stuff for it to be able to divide in the next cycle. There is the interface in which the cell's DNA is duplicated, uh, or the S phase, where the cell's DNA is duplicated, and it and more proteins are synthesized so that it has enough material to divide into two cells to generate two daughter cells. There is a G2 phase, which is again a phase in which the cells accumulate proteins, lipids, etc. They synthesize a lot of things for it to pass on to the next two cells, and then it further enters the M phase, or the mitotic phase. Just a second. Um, okay. So, as I said before, during M phase, all of these components are going to disintegrate, and they would reassemble only after the cell has exited the M phase. Uh, there is also a G naught phase in the cell cycle, in which the cell sort of decide whether it has enough resources to go through with this cycle of division. It also decides whether it is so. I don't think this has been mentioned, but anyways, so every cell has a very limited capability of being able to divide. This is something There is something known as the Hayflick limit, which basically mentions how many times the cell can divide, which depends upon there is a region in the DNA of every cell called the telomeres, the the chromosomes of the cell. So these telomeric region are basically repeats which do not code for anything, but these um, with every cycle of division, these shorten. And the Hayflick limit basically proposes that once the telomeres have vanished, the cells can no longer divide. Uh, the s- the cell basically needs to decide all of that before entering the M phase, whether or not it can undergo another round of division. Because if it undergoes more rounds of division, that without a section of the telomere present, it's going to start affecting the genetic composition of the cell. It's going to start mutating the genes. So basically, G naught is the sort of decision making phase of the cell. The others other phases are G1S, G2, and then there is the mitosis or M phase. uh, so the mitotic phase itself has been divided into different parts. There is the prophase, there is prometaphase and metaphase. Usually uh, this sort of gets blurred between prophase and metaphase. There is anaphase and there is telophase. During prophase, the chromosomes would condense. The centrioles would the centrosomes would have spindle fibers coming out of it, which would then attach to which would later on attach to the sister chromatids. The nuclear envelope would break down the further on, the organelles are going to break down. The During prometaphase, the chromosomes are still condensing. They reach their final state only during metaphase. The chromosomes are sort of uh, till prometaphase. The chromosomes are sort of placed are all over the cell, so to speak. Um, in the transition between prometaphase to metaphase, the chromosomes would align at the middle of the cell, and spindle fibers that are emerging from the centromeres uh, would then attach to these chromosomes, and then the spindle fibers are going to pull apart these two sister chromatids. So if you if there is so if this is a chromosome that uh, is going to be separated during the cell division, uh, the spindle fibers would attach here, and then they would pull these. So basically there will sorry so if you were to pull a line, sorry uh, a line through this, it's basically going to pull one centromere is going to pull this one, where the other one is going to other central is going to pull this one, to do different sides of the cell. um, then yeah then these uh, the bindings between these while due to the pulling and due to other proteins, the binding between these two sister chromatids is going to break down, and these then during the telophase, the chromosomes which have arrived at the opposite sides of the cell are going to condense again to form what was before, and there is the nuclear envelope reassembling. The organelles will start to reassemble during cytokinesis. There will be a furrow that forms between. So until telophase, the DNA has separated, but the cytoplasm is still a whole thing. It's not two separate entities. So there is this portion called cytokinesis, in which cleavage furrow would occur in the cell, and the cell is going to the cytoplasm is going to bud off into two pieces. um, there are cases in which uh, the cells would the cells would be entering M phase, but because of some defects, they can't undergo cytokinesis, or because of how the environment is, they can't undergo cytokinesis, in which case they will have two nucleus present in the cell. So basically that cell will become multinucleated. Similar thing also happens in muscle cells, in which they don't really go undergo cytokinesis. Rather, they are multinucleated. syncytium of cells. Um, Again, yeah, this is what our interface nucleus looks like. The chromosomes are all condensed and not very clearly, uh, can't be very clearly demarcated unless you are using very specific markers for it. There are euchromatin regions. There are regions which are very densely packed called heterochromatin regions. Whereas this is the interphase nucleus. This kind of nucleus is present during G1, G2, S phase. Whereas there would be the this is the pro metaphase nucleus, in which all of these chrom all the chromatin sort of condenses and forms chromosomes, and because there are 26 chromosomes in human genome, there'll be 26 condensed chromosomes present here. um Sorry, 46 chromosomes and 23 pairs. And then this would uh, they would separate in the subsequent stages and form two daughter cells. uh, Similarly, different species, as you can read, have different number of chromosomes, and this would lead to differences in the cell division, but the basic procedure remains the same. Meiosis, on the other hand, is very different. Um, this pictorially represents how cell division would occur. The Going from interphase to prophase, the organelles have started to fragment, and then the centrioles have started to assemble. They latch onto the chromosomes and then pull them apart, and then reassemble everything at the corners of the cell, and then there will be cytokinesis or division of the cytoplasm. um so the pulling apart of the sister chromatids occur due to microtubules. So the centrosome will release microtubules which are going to latch on to these chromosomes. Whereas the actin cytoskeleton has a different function. It still protects the cortex of the cell. uh, After everything is done, after all the separation of sister chromatids, everything is over. These The actin cytoskeleton is the one which is going to form the cleavage fora, and they sort of rearrange themselves and lead to the budding off of the cytoplasms, resulting in two daughter cells. Are there any doubts or something until this point? Okay, cool. um Move on. Okay. Again, cell division, segregation of duplicated chromosomes. uh, all there would be equal partitioning of daughter cells if the if it is giving rise to two daughter cells of equal identities. There are cases in which the division of the division results in daughter cells which are unequal, or they have so if sometimes a stem cell might divide to give rise to one differentiated cell and one stem cell, which the stem cell goes back into the reservoir of stem cells, whereas the differentiated cells goes ahead to perform all the functions and everything. uh, that kind of unequal division can happen, but uh, the cell division might also result in formation of two completely identical cells. Um, these two cells will also have similar genomes because they arose from one uh, mother cell, so to speak. Um, there are different proteins. So, this is what I was talking about, the decision phase, the G naught phase. The cell is basically going to see if it can undergo division in this cycle. If it can't, then there is no point of it preparing DNA of replicating DNA of preparing other proteins for synthesizing more lipids and all of that. If that were the case, it's going to exit the cell cycle and become a differentiated cell instead. This is another, this is what we have discussed. Yeah. So this is what the what the nucleus would look like. The chromosomes would look like. This is not. Uh, this piece of DNA has not condensed yet. It's still in the chromatin form, whereas it uh, as it progresses from prophase to metaphase, it's going to condense and uh, the um, form the chromosomal form of DNA, so to speak. And then the middle portion, which is what the centrosomes latch onto, is called the centromere. And the centrosomes release microtubules that is going to bind here. And all of this is sort of held together by a group of proteins called as cohesins. This is one sister chromatid and this is the second sister chromatid. uh, so depend there is also this thing that uh, each chromosome might have a different kind of structure depending upon its length, and secondly depending upon where the centromere is positioned. So there are four different kinds, if I'm not wrong: acrocentric, telocentric, uh, mesocentric or something. There are four different kinds depending upon where this is placed. Anyways, um extra information might not need. uh so yeah, uh the uh the centrosome is going to release microtubules which are going to sort of bind to the centromere, the kinetochores present in the centromere, and uh, then this would there will be a pulling force applied, and applied to both of the from both the sides. The cohesins are going to dismantle. So these cohesins are a group of, they are a complex of proteins, they are not single proteins. So these are going to dismantle, and then they are going to separate. um okay. So, this is what a centrosome looks like. There are two centrioles present in each centrosome, and this also has a nine triplet, no, sorry, nine doublet, 9+0 conformation of. So basically, these are made up of microtubules, and they have a 9+0 conformation, in which this one triplet is one. So there are nine such repeating triplets present throughout the centriole. um, so just before the cell division, the centrosome also has to divide itself to form two centrosomes, which can then go to the opposite ends of the cell, and and separate out the cystochromatids. Uh, so if this is one centrosome that's present, which has two centrioles, a mother centriole and a daughter centriole, there is no such clear distinction as to what the mother centriole is and what the daughter centriole is. This is just nomenclature based on what, uh, like, you can differentiate, because the mother centriole is sort of older. It's whereas the daughter centriole is going to be assembled newly in that cycle, and there are these proteins that are present only on mother centriole. uh, so these two centrioles are going to sort of disengage, and then new microtubules are going to come in and assemble, one attached to the earlier daughter centriole, and one attached to the mother centriole, and the proteins that are present in the mother centriole would also assemble on the daughter centriole, which is called as the uh, which is what happens during centrosome maturation and engagement. Then these two pairs of centrioles are going to separate due to the microtubules. They move away from each other, and then they form spindle. They spindle fibers arise out of these centriole pairs and latch onto the chromosome. Mm. Again, this basically mentions that the actin cytoskeleton is going to be rearranged, and there is also going to be a rearrangement in terms of how the lipid components are going to be present in both the cells. So if you are given rise to two identical cells, you basically give basically have to give them equal lipid composition as well, right? If the surface of that cell might not have uniform lipid composition, so it needs to rearrange all of that and then divide the plasma membrane as well. There is also going to be a growth in plasma membrane, so during, where is it? So during the cell growth phase, uh, the cell not only accumulates stuff for the growth cycle, it also results in a growth of the plasma membrane. It's going to synthesize more lipids which are going to sort of merge into the plasma membrane. So the cell which is going sort of going to undergo division is would be bigger than a cell which just underwent division, if I that makes sense. um Again, there is going to be a furrow that actin cytoskeleton forms, which is then going to merge with the microtubules that are present in between the uh, uh, in between the two daughter cells, and this uh, basically forms the mid body structure. And then there will be various proteins that are going to assemble at this position and twist off the plasma membrane so as to separate all the both the cytoskeletons, uh, resulting in two daughter cells. um yeah, this is what I was talking about. The cells need to regulate how their lipid composition is and how the localization is. They During the G2 phase, they are also going to synthesize more lipids, and then it's going to merge into the plasma membrane, newly synthesized, uh, depending upon whether they are supposed to be present in the outer or the inner leaflet. Um, but there are still some open questions in this area. Um, the other thing that I had mentioned previously is that the nuclear envelope also disassembles during mitosis. So there are different mitotic kinases which are going to phosphorylate a lot of stuff inside the cell. They also phosphorylate proteins that are present in the nuclear pore complex and the lamina, etc. So basically, this phosphorylation of the proteins would be a signal for their fragmentation and for their disassembly. Um, so if you modify a protein by adding something, for example, a phosphate group, the protein's conformation might change, which is what happens, and because of that they sort of lose their linkage to each other. So the uh, lamins form like filaments of filamentous structures, right? They are, because of phosphorylation, they are sort of going to detach from each other, and then they'll be floating around in the cytoplasm until the mitosis is over, and they can then reassemble and uh, form all of the structures that they were forming previously. um, the Golgi is also going to uh, going to fragment during this process. However, it does not open up, rather, it forms like the cell chops it up into smaller vesicular structures, which can be reassembled at the end of the mitotic cycle. Um there are different proteins that are responsible for uh, keeping the Golgi together, such as GM130, GRASP55, 65. Uh, the mitotic kinases that activate during the M phase that result in dissolution of the nuclear pore complex and the lamins. They are also going to uh, uh, destabilize these proteins that keep the Golgi together, and because of that, they sort of the Golgi sort of separates out, and then there is this protein complex called CTBP1 bars complex, which sort of cleaves the Golgi into smaller vesicles. Once the mitotic kinases retreat, the cells the these proteins will lose the phosphorylation and then reassemble, uh, in simple terms, and then reassemble to form the Golgi again. um One more thing is that, so if you have a adherent cell, thus when the cell is undergoing division, it loses all the contact points with the substrate, and it rounds itself up. And after the division has occurred, after the two daughter cells have formed, they again sort of uh, reattach into the substrate. This sort of rounding up also happens during the also happens if the cell is not present on a 2D substrate, rather present in a 3D substrate. So whenever the cells are present on a dish, say, in which they are not embedded in anything, this is called as 2D substratum. Whereas if the cells are embedded in different kind of materials or extracellular matrix such as collagen, uh, or fibronectin, etc., that is called as the 3D substratum. In both the cases, the cells are going to round up because of the changes that happen during changes in actomycin cytoskeleton that happen during the cell division. um okay. uh Again, mitochondria is also going to fragment during cytoskeleton. So there are these uh, mitochondria itself in a normal cell is undergoes there is a constant cycle of fission and fusion undergoing in case of mitochondria, but it's going to fragment even more during the case of mitosis, in which, so CDK1 and Aurora A are basically kinases that activate during prior to mitosis, and they uh, sort of dissociate the things that are holding the mitochondria together, because of which the mitochondria would then bud off. After cytokinesis, the mitochondria can again fuse and then be reassembled with these proteins. Um, the the proteins that are responsible for fragmentation would be degraded uh, after the mitotic cycle is over, so that the mitochondria does not keep undergoing fragmentation time and again. uh, yeah. Okay, this basically is a live cell which I can't play. It's basically a depiction of what happens during the cell division, but I can't play this. Um, anyways, um, yeah. So basically, you know uh, what the cell division is in simple terms and what happens in each stage, and what kind of structures disassemble and then reassemble uh, during my during mitosis and after mitosis, etc. uh, do you guys have any doubts up until now? If not, I can start uh, presenting the questions. Okay. Okay. um are you guys able to see the screen? Okay. Um, okay. So the first question is the complex of proteins associated with the inner and outer nuclear membrane. So if you remember, there is this link complex which uh, links the cytoskeleton to the nucleoskeleton, which is what is associated with the inner and outer membrane. The snare and clathrin complexes are uh, vesicle fission fusion. Snare is for vesicle fission, clathrin is for endocytosis of vesicles, etc., which you don't need to know. Which of the following statements about euchromatin is true? Euchromatin is highly condensed region of DNA, which is false. It is rather very loosely packed and uh, transcriptionally very active. Uh, Euchromatin is exported across the nuclear pore complex, which is false because it so the euchromatic and heterochromatic regions are going to stay inside the nucleus. It's the RNA that gets synthesized from the euchromatin regions, which is going to be exported out of the nuclear pore complex. The euchromatin is transcriptionally silent, which is again false. It is very transcriptionally active. Most of the genes that are expressed in the cell are present in the euchromatin regions. Euchromatin is less condensed and gene rich, which is true. Lamins are cytoskeletal elements present in the nucleus. The outer membrane of uh, uh, the outer nuclear membrane of the cell is continuous with the ER membrane, if you remember. Okay, let me just, if you remember, did that if you remember this image, the outer nuclear membrane, and this is the ER. Then these two are continuous. What is the role of nucleolus in cells? It's mostly responsible for rRNA synthesis or ribosomal RNA synthesis, which forms the ribosomes in the cell and are responsible for translation of the uh, components of the translation of RNA into proteins. Um, the fibers formed during the cell division are made up of microtubules, uh, and this is what latches onto the cystochromatids. Actin cytoskeleton, on the other hand, is present in the cortex of the cell. Um, during which phase of cell division are the chromosomes attached to the spindle fibers? It's the metaphase. Uh, remember what happens in what stage of the cell cycle. That is very important. The phenomenon of nuclear division is called karyokinesis, whereas the phenomenon of the cytoplasmic division is called cytokinesis. Um, which of the following statements about cell division is not true? uh, Golgi fragments and falls back into ER. Uh, true. Mitochondria chondria fragments and the fragments are distributed throughout the cells. True. DNA condenses to form chromosomes. True. Actin has no role using during cell division, which is false. It plays a role during the cytokinesis of the cell. The centrosome has two centrioles. How many microtubules are hence connected to each centrosome? So, if you remember, uh, if there are two centrioles and every uh, and if you remember, there are 9 sets of microtubules, and each microtubules that connects to the centrosome connects to one triplet. So there are 18 triplets. I hope that was clear. Are there any other questions? Are there any questions or any doubts that you guys have?
Ma'am,
Yes, tell.
ma'am, can you explain kinetochroma?
Sure. Uh, 1 second. Okay. So, um, mm, so if you know this region is called the centromere of the chromosome, right? And the centromere also has a protein complex associated with it, which is known as the uh, which is sort of known as the kind of, there is a structure here which is known as the kinetochore, and the microtubules that arise from the centrosome uh, attached to the kinetochore here. They don't. So basically the centromere is a superset of kinetochore, and in simple terms, and uh, this forms the site of attachment of microtubules from the centrosomes. Did that make sense?
Thank you, ma'am.
Are there any other questions?
Ma'am, can you explain the last questions in the assignment?
Sure. Okay. So every centrosome has two centrioles, right? And the microtubules that arise from each, there will be one microtubule arising from each triplet. Uh, so the each centriole has a 9 plus 0 conformation. There would be nine triplets that form the cylindrical structure, right? So, because there are two such centrioles and nine such triplets, it would become 18 triplets. Did that make sense?
Yes, ma'am. I get clarified.
Oh. If there are no more questions, we can end the session. No. Otherwise, if there are any questions, feel free to ask.
Ma'am, a question.
Yes.
Ma'am, is there any question bank or something to prepare for the exam? Like, is there a question bank that is there?
Um, I'm not really, but I will be sharing all of the questions that I have discussed, and I think you would have access to the questions that other TAs have shared. So that can form a question back, but you can read books such as Albert's, which are more detailed, but it will give you a good grasp on the concepts. But the questions would be like this, so you don't have to worry so much.
Ma'am, where will it be shared, ma'am?
I think you would receive a link. Not sure. I am not sure about all of the admin stuff. Sorry.
Yes, ma'am, sure, works.
Can you explain the proteins involved in the architecture
Of
and
architecture of.
the fission process, fusion process?
Okay. Um. Sure. So maybe one thing is you can go through this review that sir has used in this set, because that mostly has all of the details. But anyway, you are talking about this, right? Yes.
This and also the next slide.
Okay. uh So, this in this, so these mitochondrial are, there is a sort of turnover rate associated in which they fragment. However, they are mostly present in a fused form in a interphase cell. What happens, so when the cell enters the mitotic phase or M phase, there are these mitotic kinases which are going to be activated. Each stage that the cell goes into, there will be a different set of kinases that are going to be activated, and each of them would have a specific function. um the DRP1 protein is known as the fission protein, uh, and these there are other proteins that stabilize the mitochondria. However, the DRP1 protein sort of destabilizes the mitochondrial fuse structures of mitochondria. What the kinases do is that they activate DRP1 so that it can form a ring around the mitochondrion and sort of constrict the mitochondrion, and there will be other proteins coming into play, uh, and this would basically lead to the fission of the mitochondria. Once the and because DRP1 is active throughout the mitosis, these mitochondria would not be able to come back together. Um Once a cell exits the mitotic stage, the because these kinases are no longer active, TRP1 cannot be active as well. So the cell can no longer has this the other proteins that are going to keep it separate are also not going to be active. However, the proteins that stabilize the mitochondria will be back, and the fusion proteins will be back, and they would then fuse the mitochondria and form the network that it had before. Did that make sense? And the proteins that are involved in the fission process will be degraded. They would be synthesized only when the cell wants to enter the next cycle of division.
Thank you, ma'am.
this uh, this is also this basically is an experiment which which was to prove whether the mitochondria that how the mitochondria is getting divided inside the cell after into two daughter cells. So there are these certain damaged mitochondria that are present in the I'm not sure about what the comet tail is, but there are certain damaged mitochondria present inside the cell, and these would uh, uh, sort of redistribute throughout the cell, and the daughter cell would get parts of damaged mitochondria, and the uh, both the daughter cell would get parts of damaged and intact mitochondria. And oh, sorry, the comet tail is basically the actin sorry the comet tail is basically the actin filaments sort of coming together to redistribute the mitochondria. They are going to take damaged mitochondria from this end to this end, and they're going to take the intact mitochondria from this end to this end. Uh, Life Act, this basically is the mitochondria. This is the actin filament network that is present. So these areas that you see, these dense areas, is the uh, comet tail, whereas this is the cortex of the cell. Mito tracker basically helps you track the mitochondria present throughout the cell. uh, I'm not sure about what the experiment was, but it basically showed that damaged and intact mitochondria get redistributed throughout the cell via actin cables, and this leads to formation of the mitochondrial networks which can function in both the cells, daughter cells. Did that make sense?
Ma'am?
Yes.
Yes, ma'am. Ma'am, I have one more question.
Yes.
Actin is made up of what?
Actin is made up of globular. So there is this protein called globular actin or G actin. So
Ma'am.
these globular actins are going to come together to form these chains of actins, and these are known as filaments of actin basically. uh, there will be other proteins, I think, which was discussed in week 5 or 6. There will be other proteins that are associated with the actin network, but the basal subunit of actin is G actin.
Yes, ma'am. Ma'am, can you summarize the all videos in a
I
short form?
I think there is going to be another session sometime this week, and in that,
Yes, ma'am.
there will I'll summarize all of the videos and everything, all of the
Yes.
PPTs and everything.
To
If
go ahead for examinations
yes, yes,
because we
uh,
had attended some of the classes and we had not attended some of the classes.
okay. Yeah, there will be discussion sessions, I think, on 20th or 21st of this week,
Yes, ma'am.
and I'll basically summarize all of it together. Do you
Yes, ma'am.
have any more
Thank you
questions?
so much, ma'am. You had answered me.
Okay, thank you.
No, ma'am.
Bye.
I had. Yes, ma'am. Thank you very much, ma'am. Bye. Good night, ma'am.
Bye. Good night.